Now, before I begin, I want to make sure everyone knows that none of this is my research or anything that I've had anything to do with. A good portion of the presentation was done by this woman. Her name is Dr. Elaine Ingham, and she wrote the USDA Soil Biology Primer back in the 90s. She wrote 8 out of the 10 chapters of that book. She said something that I waited my whole life to hear in a presentation, and she said this: there is no soil on the planet that lacks nutrients to grow plants. And until you run out of rocks, you have everything there to grow plants. And why I thought that statement was so true is because I grew up on a farm next to a river, and they had a lot of sandy ground. We had some good ground, but we had a lot of timber ground. And I spent a lot of my time growing up in that timber ground, observing nature, and I would see these trees growing in the timber ground robustly each year, putting on biomass, sequestering nutrients from the soil many times greater than our best cornfield or our best soybean field. And yet nobody was fertilizing it. Nobody was putting on pesticides. But when I was doing farming, it was pounded into my head that if you grew crops, you have to use fertilizer. Whatever you take off for crop, you have to replace. The soil was like a bank, and you have to return it back in. And the way you tell how well you're doing, to know how much energy you have in that bank, is the soil chemistry test. The next person that a lot of this information is coming from is Dr. David Johnson. He's out of New Mexico State University, and he's done a lot of the replicated work that Dr. Elaine Ingham did when she was at Colorado State University, led by Dr. David Coleman back in the 80s and 90s. So first we need to talk about what is soil and what is dirt. So from the very beginning of soil science, the person who developed the whole discipline of soil science is the Swiss-born American named Hans Jenny. He was a soil physicist at the University of California at Berkeley. And he traveled all over the world classifying soils, and he took with him very good German microscopes. And when he looked at these soil series worldwide, he saw that it was not only the size fractions of the parent material, the sand, the silt, the clays, the rocks, the pebbles, it was fundamental biological groups that he saw in the soil. And that is bacteria, fungi, and the root fungi, protozoans, microarthropods, and nematodes. All of those things had to be in the soil for it to be called soil. So that was the beginning of soil science. But something happened after 1940 where we dropped off the biology and focused solely on the texture of soil. So what we did is we replaced those organic matter and those organisms with inputs. So there is a difference between soil and dirt in terms of its function. Soil organisms build the structure. In soil, bacteria and fungi build soil structure. Nothing else on this planet builds soil structure except for bacteria and fungi. Nutrients are held in the soil. They're held in the bodies of the bacteria and fungi in the organic matter. Water is retained in the soil, and it moves slowly and does not run off. And the water that's going through soil is completely clarified. It is drinkable. If you have water running off your farm and you can't drink it, you don't have soil. So when we talk about dirt, we have no organisms, we have no structure. And that's not necessarily true. You do have the organisms. You have galaxies upon galaxies of bacteria building microaggregates, and we're going to see that. But you can't build soil structure without fungi. So you don't have the soil structure. Nutrients move with your water. Water is not held in the soil pores, it moves out. So not only are you drought prone, but now you're flood prone. One of the things here at Lauren's farm, when I was driving in, I had a feeling, I had the sense that Lauren is working with nature. And to hear that is a trout that is naturally reproducing in your stream makes me understand why, because he is not leaking any nutrients into that water. If you leak nitrate into that water, you won't have the trout naturally reproducing in that stream. Because we need to be clear when we're talking about nitrates in water. Nitrates are toxic to all life forms on this planet except for 3 organism groups: bacteria, fungi, and the root systems of plants. To all else, it is toxic. Now, we're talking a lot about microbes, and that's going to be my whole presentation is on the microbial life in the soil. Microbes are the backbone of every organism and every ecosystem on this planet. Living organisms have shaped almost every facet of our planet's development for the last 4.2 billion years. So right off, just in the beginning, I'm talking about the fungal to bacterial biomass ratios in the soil and why it's so important. Well, the main reason why it's so important, because bacteria and fungi were the first to come on this planet, to develop on this planet. Around 4 billion years ago, bacteria appeared on the planet. And then approximately 3 billion years or 2 billion years later, fungi appeared. And all the other organism groups on this planet radiated out of the fungi. And those organisms groups had to make relationships with the bacteria and fungi because only the bacteria and fungi have the genetic material to solubilize all of the 99 naturally occurring elements in the periodic table and make them available to all the other living groups. So every atom that's in my body and all of your bodies have come from bacteria and fungi expressing their genes and pulling from the crystalline structure, cobalt, zinc, all of those 99 naturally occurring elements and make it available so we can take it into our body. So what builds soil structure? So what builds soil structure is bacteria and fungi. Bacteria make the fundamental unit of soil structure. It's called the microaggregate. Right here, that is the microaggregate, and we're going to look at that on the microscope in Bob Recker's lab, outside his mobile lab. So that is a microaggregate, and it is only built by bacteria. In that clump, there's approximately 500 bacteria. So when bacteria eat food, when they eat carbon resources that are provided to it by the plant or organic matter, they produce sticky glues as their waste products. And they stick themselves to a sand, silt, clay, and they hold it together in space. So that is the microaggregate, or the most fundamental unit of soil structure is that. Now, Dr. Elaine Ingham has a good analogy of soil structure. She has what we call the brick house analogy of soil structure, where the most fundamental unit of a brick house is a brick. So the most fundamental unit of the soil microbiology is the microaggregate. But you can't build a house out of bricks alone. You need something to glue those bricks together. Fungi grow in long strands around things. They basically will lasso 5 or 6 microaggregates and make a macroaggregate you can see with your eyes. At the same time, making hallways and passageways so air in your roots and water can move deep down in your soil. Now that is just the structure of soil is made by the bacteria and fungi. Bacteria and fungi are also the power generators and the transporters of nutrients in the soil. So think of the bacteria as the power plants of the soil. Nothing else on this planet generates more power than bacteria. But there's no good to have a power plant that doesn't have transmission lines to take the power out to the soil. That's where fungi come in. Fungi are both the transmission lines and the communication system of the soil. So I need to talk more about fungi. Fungi, when I first heard about fungi and I got into this, I always thought of mushrooms. But mushrooms are the fruiting body of fungus. They're kind of like an apple on an apple tree. The vegetative form of fungi is as large as the forest itself. Fungi grow in long strands through the forest around things, holding the soil together. And they can get quite large. The largest organism on this planet is a honey mushroom in the Pacific Northwest that is approximately 3,000 acres large and goes into the soil about 15 feet deep. So fungi provide both the structure in the soil, they provide the logistics and the communication in the soil. So the logistics, they're taking resources and allocating it out through the soil. They're making decisions on what organisms get what nutrients and what plants get what nutrients. Now, if we think about, for instance, if we go to war with a country, what are the first 2 targets we take out when we're going to war with a country? That we take out the logistics and we take out communication. Right now, what we do in modern agriculture very well is we kill off all the fungi in the soil. And it's not because we don't understand it, it's because we can't see it. But if we look at the soil with the microscope, it will become plain to you that's exactly what we're doing. So when you look in the soil and you look at the soil, at the fungal structure, you start seeing these patterns. It's called the mycelial archetype. And this mycelial archetype is a basic pattern and structure that's found throughout nature. What was really interesting is astrophysicists were coming to soil microbiologists and people who study fungi, and were asking them questions about the fungus in the soil. The reason why they were asking them is the way the fungi looks in the soil is the same way that dark energy is distributed in the universe. It's the same way that matter is distributed in the universe. Fungal mycelium looks like neurons, like our neurons. Fungi have this general shape and pattern. And fungi communicate just like a neuron. This has just been discovered. People who study fungus were wanting to understand how fungi could communicate across many, many distances without so quickly. And what they found out is that fungal hyphae function just like neurons. They have the ability to transmit electricity. And when they started to test it, they were seeing all kinds of electrical pulses going through the fungal mycelium. Because we have taken out the logistics, because we have taken out the communication system in our soil, we are now in energy deficit with our corn, our soybean production, all of our production we have in agriculture. This is energy returned on energy invested. So in 1940, for every calorie of fossil fuel we have put into the soil, we got 2.3 calories of food from that. Now fast forward to 2015, for every calorie of fossil fuel we put in the soil, we get 0.1 calorie of food. Or another way to think of it, for every calorie of food that we get from agriculture, it takes 10 calories of fossil fuel. So agricultural production is actually deduction. It's deduction because we have killed off the fungi in the soil. So this is a chart that Dr. Johnson did, and it showed the fungal to bacterial biomass ratios of different soils and the productivity you have once you increase the fungi. So when you have a fungal to bacterial biomass ratio of 0.01, you basically have no fungi. So with that type of system, the energy into the system and the energy returned, you only get 3% back of what you put in. Our current agricultural systems, we're getting 11% back of the energy we're putting in. Now look what happens when you increase the fungi in the soil. You get 56% of the energy that you put into the soil, you get back in the form of carbon biomass. So right now in agriculture, we are at this phase right here. We're in this phase where we have no fungi or very little fungi. Now, the fungal to bacterial biomass ratio, that determines what plant groups grow where. So when you have no fungi in the soil, you have set the stage for different plant groups that have bacteria being their microbiome. So weedy species that we're currently trying to kill, their microbiome is completely bacterial dominated. They get all the nutrients they require when the soil is bacterial dominated. And that's why they're growing so robustly. We can't kill them. Now, once we get the fungal component back, the weedy species change. It's like succession in an ecological system. So for corn, soybeans, most of our row crops, we need an equal amount of fungi and bacteria. So what we need to do is figure out how we can get the fungi back, and most importantly, stop killing the fungi. So right here, this is our state in succession. We are at early successional state. It's basically, in nature, this is like a natural disaster. This would be like after a flood or earthquake, something like that, where we've killed off all the fungi in the soil. The soil has been mechanically tilled. And the weeds come in after that because their microbiome is bacterially dominated. Now, weeds have a purpose. Their role is to come in, cover the soil quickly, and produce biomass. That biomass is food for the fungi that will start to move up. So when the weedy species come in, they have a robust growth, they die back, and they provide food for the fungi, and you will increase the fungal biomass as succession takes place. So what we need to do as agricultural producers is to get that fungal biomass to equal amounts of fungi and bacteria right here. And we'll talk about ways to do that. Now, this is interesting to understand too, that the forms of nitrogen in our soil are controlled by the bacteria and fungi. So, for instance, to have nitrate in your soil has to be completely bacterially dominated. Or in another way of saying it is when nitrogen is mineralized. So, for instance, if you're releasing nitrogen in your soil, it always goes through the soil microbiology. So when a bacteria eats food and grows and gets eaten by its predator, only then is that nitrogen released back in the soil. So when it's released, it's always released in the form called NH4 ammonium. It's only when that soil is dominated, or that microsite is dominated by bacteria, will that ammonium be changed to nitrate, because the nitrifying bacteria can pull off those hydrogens and replace them with oxygens. So if you have nitrogen or nitrate in your water, you already know your soil is bacterially dominated. So for instance, if you have that same mineralization of, say, a fungi being eaten by a fungal-feeding nematode, and when it releases that waste from eating that nematode, that NH4 will stay as NH4 in that microsite if it's dominated by fungi, because fungi will change the pH of that microsite to be slightly acidic. And the nitrifying bacteria can't express their nitrification genes to take off the hydrogen, so it will stay as ammonium. So for the human body, we have 200 human genes. We have 2 to 20 million microbial genes. So we have our own microbiome as well. And it's not only that, we have 20 trillion human cells. We have over 100 trillion microbiological cells. And we have over a quadrillion resident viruses. Now this is kind of shocking because one virus, as we know, has kind of brought everything to a standstill. So what we need to do is we need to change the way we think about the soil. The soil is a system. Instead of doing what we have done with science, where we think of reductionism, where we break things apart and study the pieces individually. What we need to do is start studying the system. So here's an excellent example of a systems approach, or a reductionary approach to systems thinking. So when we talk about the soil has galaxies of microbes all interacting to each other. It is unbelievably complex. But with a reductionary approach that we're currently using in science right now, we're taking one or two things and studying them alone in isolation and making a conclusion. The problem with that is they all work together as a system. So to give you an analogy of how that gives you how it gives you an understanding that's not representative of what is happening in the soil, is think of a bicycle. A bicycle has 600 and some parts. Now, if we're doing a reductionary approach, we're going to break apart those parts and only look at a couple parts. And imagine we can't see the whole part of the bicycle. We can only see 2 parts. Now, if we studied a spoke and a chain ring, it would be very hard or impossible to understand. When we put all those parts together, we get an emergent property of transportation. Because studying them alone in isolation, you don't. So the system approach is what we need to move towards when we look at the soil, since it's so incredibly complex. Now, a good example of this reductionary approach and how we can be led astray is this work done by Richard Mulvaney out of the University of Illinois at Champaign. Now, he is a soil fertility specialist, and he wanted to understand what happens when you put on inorganic nitrogen to soil organic matter. You know, when you put on inorganic nitrogen, there is a lot of bad things that are associated with it, like we talked about nutrients leaking out of the soil into your water. So one of the things that the agrochemical industry and the reductionary approach to understanding this said is that even with all of those problems, nitrogen will increase the yields and therefore increase organic matter in your soil. Organic matter is so important because it controls 80% of your soil function. It also gives you fertility in the future for many years in the future. So applying inorganic nitrogen actually increases your soil organic matter. The problem is that no one ever tested to see if this was true. So what Dr. Mulvaney and his team out of Illinois did is they looked at long-term data sets throughout the world, and they looked at what happened when they started using inorganic nitrogen. So for instance, in the Morrow plots in Illinois, they had continuous corn data from the 1870s. So they could see what happened to the soil organic matter once they started using inorganic N. And when they looked at the data and they did the proper controls, what they found out is that the soil organic matter tanked. The soil organic matter started to go down dramatically. And this is not no-till, this is moldboard plowing. So they looked all over the world. They went to one of the longest term crop production studies in the world at Rothamsted, England. And the same thing happened there in the 1800s when they started using bird guano. So what they found out is when they looked at the data is that what nitrogen does to your soil, it never really actually gets into your plant. What happens is when you put on inorganic nitrogen, the bacteria eat all of it. You have galaxies of bacteria that will eat every ion of that nitrogen. And then once they eat that nitrogen, they can eat carbon that's in the soil. So you explode the bacterial population in your soil. It is only when they're eaten by their predators is that nitrogen released as inorganic nitrogen. So when you add inorganic nitrogen, you are essentially burning up your carbon in your soil. So Dr. Mulvaney does this analogy. Inorganic N is to your soil organic matter as lighter fluid is to charcoal. It allows you to better burn it. And this is not a new understanding. A longtime researcher back in the 1920s, William Albrecht, he reported this at Sanborn Field in Missouri back in the 1920s, where they were doing continuous wheat and putting on inorganic nitrogen. And his conclusion is the soil organic matter was burned out by putting on excessive amounts of fertilizer. This is another example. This is Gabe Brown's soil. When he started to do no-till in 1993 and doing cover crop diversity, once he reached that 3% organic matter, that's when you have the ability to hold on to microorganisms. So the microorganisms need a home. 3% organic matter is the baseline for these organisms to have a place to live. Otherwise, if you have anything below that, they're not going to be coming back the next year. So once he did cover crops and started doing multi-species and he stopped using inorganic N here, he started to build carbon in his soil, especially when he started to use multi-species and integrate livestock. So this is a study, a replicated study that David Johnson did on what Dr. Elaine Ingham did back in the 80s and 90s at Colorado State. So they actually did a test to see whether nitrogen, phosphorus, potassium, organic matter had any correlation with plant growth. And if we look at nitrogen, there's no correlation between plant growth and nitrogen. If we look at phosphorus, there's no correlation between phosphorus and plant growth. And potassium, no correlation. And this one's kind of a shocker. There is no correlation between organic matter and plant growth. But there is a strong correlation between the fungal to bacterial biomass and plant growth. So here are the plants that he used in that study. And you can see as the fungal component increases in your soil, you get robust plant growth. These are pepper plants. Now, the best way to tell how you're doing in the soil for the fungal to bacterial biomass ratio is with a microscope. Now, once you get to start using this microscope, it doesn't take very long to do it. Give me half a day and you will be able to do your own assessments in your own field. And you will know within minutes what your fungal to bacterial biomass ratio. So it doesn't take long. It takes maybe a half a day to get training on how to do it, but you'll be able to assess your own soil right away. And literally it'll take minutes to see where you're at in the succession, and where you're at in succession on your farm. And if you can grow your crops without inputs. So this is what we're looking for. This is a beneficial fungal hypha strand in the soil. They have definite characteristics, and you can tell right away if it's a good guy or a bad guy depending on its width and its color. But this is what I have seen in hundreds, literally hundreds of soil samples I've taken throughout Iowa. It is all bacterially dominated. There is no fungi at all. And like Elaine Ingham says, if all you have growing in your soil is weeds, if you didn't do anything to your soil and weeds come up, you already know what the fungal to bacterial biomass ratio is. It's all bacterial. So this is my garden. I finally got my garden to a fungal to bacterial biomass ratio to 1:1. It took 5 years to do it. I made a lot of mistakes. What that means is if you have a 1:1, that means when you're at 400x, you have 20 fields of view. And you go through each field of view at 400x, you should have a beneficial fungal hypha strand in each field of view, and that's 1:1. So what I failed to talk about for this fungal to bacterial biomass ratio is that the bacterial component in your soil stays the same throughout succession. You need around 2 billion bacteria all throughout succession. What changes is the fungal component. Some of the most interesting systems research in the world is coming out of Germany, out of the Jena experiment. And they are doing things that are really interesting. They're doing this long-term multifactorial study, all kinds of research done to the nth degree like the Germans are really good at. And one of the things that they did is that they did a comparison of 8 to 16 species grown together with 0 nitrogen, and they compared that to 1 or 2 species grown with 200 units of nitrogen for 15 years. And what they found out is the 8 to 16 species out-yielded the 1 or 2 species with 200 units of N for a straight 15 years. Not only did they out-yield, but the soil organic carbon increased dramatically. The soil microbiological diversity increased dramatically as well. So what they theorize is that because you had 8 to 16 species grown together, you had such a diversity that it attracted pollinators to the site. Like we talked about, every organism on the planet has their own microbiome. So when they went to the site to pollinate the flowers that were growing together, they shed their microbiome and in essence inoculated that soil with good biology. And then you had snakes, birds, other critters would come in there and leave their microbiome and increase the soil microbiological diversity in their soil. So they have a website and they have a clearinghouse of their data that they release every month. It's unbelievable. So I highly recommend checking out the Jena experiment. So everyone's heard of the 3 sisters. So to grow corn, you grow other plants with it. And if you had any trouble, you added a little fish. So this enters this guy right here, Bob Recker, who's right over there. I was asked to work with them on soil microbiology, and one of the main reasons why I wanted to work with them is because they're doing wide row corn with 20-some species of companion crops. So that's not the 3 sisters, that's like a family reunion for the soil. So based on the research in Germany, that said 8 to 16 species grown together out-yielded 1 or 2 at 200 units of N, I have a feeling that this, with no nitrogen, is going to be the way forward for growing crops. Not only are you able to grow corn and hopefully get the same yield without adding inorganic N, but Bob is also using fish. He's using liquid fish. instead of burying a fish like the Native Americans would do. The other thing is what you're doing, Lauren. I mean, this is absolutely fantastic, where you're doing relay cropping with rye. When I pulled in and I saw all the soybeans there that had the rye harvested off, all I could think about is why on earth NRCS is not paying people to do this. It's a brilliant idea. So this is the next way to think of getting biology back to your soil, is by growing more plants together. That's what nature wants. So if we go back in history, a long time ago, we go back to see how did we grow plants, say for instance in the Roman Empire. So what we do when we're growing plants back then, you didn't have fertilizer, but you made compost. You made compost tea, extract. So you took materials and broke them down and had made an inoculum of beneficial microbes for the soil. So the oldest known book of agriculture was written by this man. His name is Marcus Porcius Cato. He was a Roman general and senator, and is considered the first agronomist in the Western world. In his book on agriculture, the first page is all about how to make compost and how to make compost tea. This is George Washington's 6-bay composting yard in his farm in Washington, D.C. So George Washington knew he would not put any manure on his cropland without it being composted. So think back in history where you don't have power equipment to haul manure away. You basically put manure on the same spot over and over. If you did that, you would essentially destroy the fertility in your field because that manure is salty. So to get rid of that salt effect, they did this composting, which is ancient. And it's a 6-bay composting yard because that's the number of times that you need to turn compost. No more, no less. If you turn it too much more, then you really don't have the beneficial organisms in the compost. If you turn it too less, then the bad organisms in the compost will take over your pile. And this is a really interesting quote that he wrote to a letter. Is that when I speak of a knowing farmer, I mean one who understands the best course of crops. Above all, Midas-like, one who can convert everything he touches into manure as a first transmutation of gold. One really good way to get the biology back to your soil, if you don't have the fungi, is to make compost. So what you want to do is to make fungal-dominated compost. To do that, we do a recipe that is ancient. This is around 10,000 years old. The Chinese have wrote about this that long ago. And this is how we make a thermatic pile. Now, what you want is 60% of that pile to be wood chips, 30% of that pile to be green material, and 10% of that pile to be high nitrogen. And you need that pile to be at minimum 1 cubic yard. And you turn that pile, and then Alicia is in this picture as well. It helps to have the new people in your office help with the turning. Mix it all together, and then you don't have the beneficial microbes in the soil. That's not, or excuse me, in the compost materials themselves. No way do you have it in there. It's been destroyed long, long ago. So what you need to do is you need to go out into the forest and find really good biology. So you want to go out to a forest that has not been manipulated, has been left alone, and has an Ohorizon. O for organic. So you'll know if it has an Ohorizon because you can shove your fingers into the soil and it will go up to your knuckles. So you just take a pinchful of that Ohorizon, and that Ohorizon is absolutely filled with the unbelievable microbiology. The Ohorizon is Mother Nature's potting soil. And we don't have an Ohorizon in Iowa. All of Iowa, the Ohorizon is gone. It's the first to go if you do any tillage, any type of manipulation. So you take that Ohorizon and you look for any fungi that is in mushroom form that is sporulating. You take a couple pieces of that, and then you look for what the Forest Service calls a Class 4 log. That is a log that's laying on the forest floor that is basically turning into soil. And you'll know if it's a Class 4 log because it will be completely wet. And right now, we don't have a lot of rain. And if you go find one of these logs, they will still be completely soaked. What's making the moisture is the fungi that's eating that wood. It's pre-wetting the wood so that it can eat it. So you take a handful of that material and you put that in the center of the pile. Those organisms will radiate through the pile and you will grow those microorganisms. In approximately 20 to 30 days, you will end up with really good compost. All the foods will be eaten and you will be left with a lot of diversity of organisms that are now dormant. They're not active anymore. And once you start making this compost, you'll have a lot of other critters start coming to your compost pile because it's such a rich food resource, mainly for microarthropods. And that's why you have wolf spiders taking over your compost piles, especially mother wolf spiders. They feed heavily on the microarthropods that are eating the wood chips or eating the fungi off the wood chips. So I will have compost piles completely covered. Every square foot has a mother wolf spider with her young. Now, when you have that compost, now you need to inoculate it into the soil. And that's where you want to make a compost extract. So you're extracting the organisms in a water bath, and now you're going to spray that extract onto your farm field. So you can take a bucket and take maybe like a pint of compost will get you 5 gallons of compost extract. You can take maybe like a half a gallon and that will give you 30 gallons of compost extract. Or you can size it up like the one we have out here today for the outside, where we're going to make 250 gallons of compost extract using a tote, 250-gallon tote. And 5 gallons of compost will yield you 250 gallons of extract. Now, how much extract you put on your soil is dependent upon what you want to do and how good the extract is. So at 20 gallons an acre is the standard application rate, so that'll give you 12.5 acres to apply this compost extract. Now you can spray it like we're talking about, but by far and away the best way to put on the extract is right with the seed when you're planting your corn or soybeans or whatever you're planting. And then you can spray it on later. You can actually do a surface application of compost, but you're looking at, at most 400 pounds an acre. You could put more on if you want, but this is like a white sheet of paper with an application rate of 400 pounds of compost an acre. It's basically a dusting. Now you can, with that compost, you can make biochar. Now biochar is something that is really exciting because this is a way that we can deal with climate change, deal with the excess of CO2 in the atmosphere, and do the opposite of the fossil fuel industry. So when we're making biochar, we're taking charcoal. So we're making charcoal, and we're taking that charcoal and we're putting it into the soil. So charcoal is recalcitrant. It can stay in the soil for 2,000 years. So for every pound of charcoal you have is worth 3 pounds of CO2 because you've blown off all the other elements on there and you're left with pure carbon. So if you put that in the soil, it's worth 3 pounds of CO2. Now, if you inoculate that charcoal with beneficial microbes by putting that charcoal in compost, the microbes go into these little structures that used to be the plant cells, and they take up residence in those plant cells for 2,000 years because the bacteria and the fungus and some of the other microorganisms are immortal. They don't die of old age. So when they take up residence in those microstructures, they take up residence for the full 2,000 years. So this is kind of like a coral reef for the soil. You put that biochar in the soil, now you've sequestered 3 pounds worth of carbon, of CO2, and that's the first level of sequestration. The next level of sequestration is now your plants grow better with less fossil fuels, less of fossil fuel inputs. And that's your second level of sequestration. Now that second level of sequestration occurs for a full 2,000 years. So this is a really good way for us to deal with the climate issue we have right now with carbon dioxide in the atmosphere. And it also is a fantastic way to inoculate our soils with microbiology.